Hi, everyone. It's a pleasure to be here, and thank you, Lucia and the team, for having invited me to give this talk on the science of longevity. I'm going to talk about 3 independent features, and I'm hoping that I can cover all of them in 20 minutes. These are my disclosures. I am also aside from being an academic, and I'm an entrepreneur. I have 3 companies, and I consult for many others. I'm going to talk about health span, longevity, progression of aging, from biological aging all the way to functional aging and decline. Measuring these things, aging clocks, the Stanford 1000 Immunomes project that I've been leading for over 15 years now. Functional aging and ways to accelerate aging research using space and microgravity, and I'm going to try to convince you not to go to space. So some definitions here. Longevity, all of you know, is extending lifespan. So this is the time between birth and death. And the origins Latin longa evus, which basically means long aging. But that's not what we really care about. That's a result of extending health span. We're focusing on mechanisms of aging and on the wellness and prevention side of things, as well as diagnostics and therapeutics. Mechanisms of aging, hallmarks of aging, biomarkers of aging, to be able to bring healthy living for a longer period of time and don't waste and lose so many years that are so precious on Earth here. And ultimately, what we want to achieve is extending quality and WALI, this concept of quality-adjusted life years and wellness-adjusted life years. And we do so by these 2 major sectors, wellness and prevention, as well as diagnostic and therapeutics, to ultimately compress morbidity, right, or extending health span. The idea of compression of morbidity was put forward by Jim Fries in early 2005, 2008. So I just want to say that wellness and prevention are really great business. The ROI is about from 4 to 5x. The global market in 2022 was 5.6 trillion. It's a huge market, and it keeps growing and growing with an annual growth of about 7%. And all these companies I'm listing down here, they reached out to us for many different collaborations. And they're trying to incorporate and develop new products and services to tackle wellness and prevention. Not necessarily because these are good people, but rather because customers are demanding these things, right? For example, Philip Morris now is working on vaping supplements and stuff like that. PepsiCo is developing different functional foods and beverages. And so in the therapeutics and diagnostic area, there's been a huge amount of investment, right? I got this slide from Phil Newman. Very kindly, he shared this with me. We have yet to see returns, as Aubrey alluded to. And so aging and longevity are great businesses in general. There's a lot of money being poured into this, but not just for investors, but also governments. We're spending over 90% of the global health expenditures here in the US in chronic diseases that are preventable. So it's a good business also for global economy. And in that realm is that we hired at the Buck Institute 2 experts in phenomic health and prevention, Lee Hood and Nathan Price. And we just announced the formation of the Human Health Span Center at the Buck Institute. Many things change with age. I don't have to convince you. Right? Gray hair, wrinkles, that's all apparent and external. Biological signs of aging, skin collagen goes down. There's social changes. You get more income as you grow older. Social connections typically go down. There are changes that are nonlinear. There are changes that are exponential. There are changes that are more linear, like visceral fat that leads to changes in body composition. And people have claimed that aging is complex, and I hate this statement because that means we don't understand it, right? I don't think aging is more complex than a function of a cell with genes and proteins and metabolites interacting to execute a function. I believe everybody in the field of aging agrees today that aging is indeed hierarchical. There is biological aging that occurs early in life with changes at the cellular and molecular level. Those are the hallmarks of aging that probably all of you know. There's tissue and organ aging. That's more phenotypic. That's what you can see in an MRI or CT scan. And then, very later in life, you have systemic changes, which is functional aging, loss of intrinsic capacity, disability, frailty, and stuff like that. These are the hallmarks of aging. We probably don't have to go through each one of those. Phenotypic and functional aging is exemplified in this figure here. I'm plotting gray matter volume that shrinks with age with huge amount of variability, which we love, right? We're embracing that variability. This is in collaboration with Joel Kramer at UCSF. But you can, this is phenotypic aging. This is what you can see in an MRI, right? But you can also see functional changes. So at the very bottom of the slide, you see mild and pre-mild cognitive impairment, and that is functional aging. So the progression of aging goes from biological to phenotypic to functional aging. How do we measure biological aging in the first place? Biomarkers of Aging Consortium is a big thing that we try now to identify and democratize and try to standardize different measurements of aging. I'm sure everybody knows about epigenetic clocks. Those are the first clocks that appeared in 2012 or so. They use CPGs that are areas in the DNA that get methylated, and there's a huge amount of different CPGs in the genome, in the DNA. And using AI machine learning, we build models to predict chronological aging, and deviations from that indicates mortality and diseases and so on. In general, the growing field of clocks have produced epigenetic clocks, have produced metabolomics clocks, proteomics clocks, cellular clocks, you name it. Epigenetic clocks are very good at predicting chronological age, but we don't understand very well the biology, and there's no very clear interventions to change and to move the needle. Metabolomics clocks, we know a little bit about biology, but not so much. It's very difficult to intervene. Transcriptomic clocks, these are gene expression, and there's a better understanding of the biology. Proteomics clocks are made out of multiple, maybe hundreds of proteins. We understand proteins much better than epigenetics changes, but it's really hard to move the needle when we're talking about hundreds of proteins that made a clock. Cellular clocks are a little bit better at understanding the biology. We know the cells very well. And the inflammatory clock that we developed, I think this is made out of 5 proteins. We understand them extremely well, and there are interventions for them. The way we built the inflammatory clock was using the 1,000 immunomes data set that we built over the years. We started in 2007, and it's been going until up until today. We secured over 70 million from NIH. We measure everything under the sun, multi-omics, et cetera. We use AI and machine learning. I'm sorry, the slide didn't come very legible, but we built an inflammatory clock for aging that tracks multimorbidity, frailty in individuals that went into the study, immune function, ventricular remodeling. This is the heart health. And in centenarians, we see that inflammatory age is much lower than calendar age, and we also predict mortality in independent data sets. We brought that to the industry. We created a company called Edifice Health. It's a whole leap to bring things from academic research to industry, but we were able to do so, and we're going to launch early in 2025, so next year. Now I'm going to shift gears and ask the audience. I want to ask everybody, raise your hand if you believe that aging should be considered a disease. Okay, that's hopeful. Great, but in the aging field, it's been really hard to convince regulatory agencies to recognize aging as a disease. It's been a long debate here, and luckily, in 2022, the WHO recognized for the first time age-related decline in intrinsic capacity. I'm going to allude to what I mean by that. In the International Classification of Diseases, ICD-11, under the code MG2A. This is really great because now we can take different interventions, bring them to the FDA, open that dialogue, and hopefully get approval and reimbursement codes, CMS, to address age-related decline in intrinsic capacity. How do we measure this thing? Very, very simply, taking different tests that are standardized. We do this in collaboration with Bruno Vella in the University of Toulouse in France. We have an ongoing study of 1,000 people that we've been following now for 4 years. It's a 10-year follow-up study that we've drawn from the iCope app that we have successfully enrolled 50,000 people in the app, 1,000 people to do all these measurements in the clinic as well as multi-omics. And my postdoc, Matias Fuentealba, really great guy, help here producing an epigenetic clock for intrinsic capacity. Okay, so for the first time here, we're talking about a clock that is not trained on chronological age. We're not trying to predict aging, but we're trying to predict age-related decline in intrinsic capacity. So ICD-11 recognized as a disease. We know that there's sexual dimorphism in the rates of decline. Females and males have different rates, etc. And very interestingly, when we compare the CPGs against first and second generation epigenetic clocks, there's almost no overlap, which means we are discovering new biology of aging. This is a pretty busy slide, and I'm just going to give you the highlight. We've been measuring this in 35 independent data sets with blood, saliva. We're doing as well as with blood, so this is a test that will come out to the market hopefully pretty soon. And it tracks immune health and mortality. So one of the things that we did was to use a supermassive language model to create different gene sets for the different hallmarks of aging. And we can see that high intrinsic capacity, or rather the declining intrinsic capacity, is correlated with chronic inflammation, cell senescence, altered intracellular communication, and not so much with other hallmarks of aging. We see that tracks immune function, as I said before, the number of naive T cells and so on, and expression of genes that participate in the immune system. We're doing much better than other epigenetic clocks at predicting mortality in independent data sets. We can also track this with health behaviors. Okay, so people taking flavonoids have much better intrinsic capacity. People taking omega-3 fatty acids from animal sources, not from plants, are doing much better as well. And if you do not adhere to the dietary guidelines for, let's say, sugar consumption, you're going to lose intrinsic capacity pretty quickly. That's what we see in this study. Okay, the last part, I think I'm going, well, this is 11 hours, 28 minutes. I'm a little bit lost here with the timing, but yes, if someone can indicate. The last part I want to touch upon is leveraging space biology. And this started with collaboration with NASA. A researcher in the Human Research Program in Houston reached out to me and said, hey, David, you know, your research in the immunome and aging could help astronauts develop countermeasures. We're developing countermeasures for space flight and the risks in cell-accelerated aging. That was shocking to me. We started a whole program 4 years ago. Unfortunately, the researcher suffered from terminal cancer. She passed away. But I took the torch and continued to do that. Many of the things that we discussed with changes at the level of brain aging, cardiac aging, immune aging. Okay, and that's what we're focusing. We published 5 papers, all came together in the Nature package in June of this year, and we discussed at length the different acceleration of aging in these hallmarks of aging at the molecular and cellular level. And so what I did was, let's try to envision a way to simulate microgravity on Earth. And can we use different blood samples and organoids that are mini organs, right? Mini hearts, mini brain. These are 2 or 3 millimeter in diameter. Can we accelerate the aging? Why do we want to accelerate the aging? To try and use different interventions and compounds, and similar to what Aubrey was doing, combinatorial compounds to see if we can reverse and prevent those aging processes from happening. And we also have functional changes in these organoids. And so we did all that. We see that there is an age acceleration in brain and heart organoids by about 5 to 10 years biological age. We can map different hallmarks of aging. We can map that to Parkinson's disease. We have a model, a very well-established model for Parkinson's disease now that we can, in 24 hours. And we can start looking for different interventions. Dilated cardiomyopathy is the disease that we're mimicking in this process. And so we started a company. Not because I like to pitch VCs, not at all, but rather because I want to bring this to the marketplace. And so we're having two applications of this technology. One is in the precision longevity field, covering wellness and prevention. And we developed the older twin artificial aging technology. It can take cells from any of you and have a window of your next 5 years of aging and see which hallmarks will affect this individual versus the next one. And we also have different recommendations as far as compounds and exercise modalities. And that comes from a large database where different exercise, physical exercise, were tested, and gene expression can change. And so let's say gene A goes down with high-intensity training, and turns out that your future aging also brings that down. So I can recommend different interventions based on this technology. And the other one is drug discovery. And so we have two models, well-established, Parkinson's disease, cardiomyopathy, and we're looking at immune aging now, vaccine efficacy, and then we're going to move on to obesity in 2026, early 2026. How am I doing with time? I think I'm done here. Thank you so much. We started this company, and thank you, everybody. Happy to discuss later on. Thank you.